right. Yeah, great. Lovely. It's wonderful to be at a conference on beyond sequence alignment, so I'll talk about sequence alignment. But really, see, I think this will show. So I will briefly touch on a couple of different sequence alignment problems. See if I can figure out how to make this work. Nope. And then just a few words about information theory at the end. The last time I gave a talk with two parts. The speaker following me took up part two of my talk and was going on, and afterwards she told me, that was wonderful. You talked about the old-fashioned stuff and I talked about the new stuff. And so I took that to realize that, of course, like everyone like me goes to sleep in the middle of someone else's talk intellectually. So by design, this was supposed to be a brown background, but the PowerPoint to PDF didn't allow the color, but you'll see my point in a little bit. I'll try to yell when I go to the next step. So reading DNA sequences, we did something very different before next generation sequencing than afterwards. And I think it's not necessarily widely appreciated, but Fred Sanger, when he first invented his sequencing method, looked around for somebody to write a computer program to put them together, even though he wasn't particularly a fan of computer programs. Michael Levitt turned him down, and Roger Staden actually took it up. And just to remind anybody who doesn't know it, the sequence assembly problem is that you get random reads located on the genome, and the goal is to find reads that overlap enough that you can convincingly reconstruct the underlying unknown text. So I'll come back to this in just a moment, the computational aspects of this. And you know, when I have a class of freshmen and I give them this problem, the wastebasket full of random reads, they think of Staden's method. You look around till you find something that overlaps convincingly, you scotch tape it together, and you keep going. That's approximately what Roger did, and later we did more sophisticated versions of that. From a computer science point of view, this is actually a hard problem. If you forget errors and everything else, and you ask for the shortest sequence that contains exactly all of the reads somewhere in it, that's actually an NP-complete problem. But the Roger Staden method would be, oh, you find a couple of reads that overlap a lot, glue them together, find another one, and go on. Just recursively get this. That this is a hard problem, you all probably know. The sequence reads have errors. Genomes have lots and lots of repeats that are of different degrees of identity. There are gaps, there are variations in coverage. And the Celera Human Genome Project had almost 27 million reads, an average of 550 bases. So the more refined version of Roger Staden has three steps. The overlap step is you look at conceptually all pairs of reads and see, do they overlap, or does one of the reverse complements overlap, if you don't know which strand the read is from. So this, if I was talking about the Celera project, would be 27 million choose 2 times 2, so 27 million squared conceptually. And then when you have all of these overlaps, which we can do faster than I just described, all of these overlaps, you look for clumps that are overlaps that convincingly agree with one another to get contigs. And finally, when you've got these guys arranged, you look down the columns and infer the letters of the sequence. I hope I briefly convinced you. The first problem is a hard problem. The next two are much harder computationally. So we were right kind of at the limit of it. The Celera Genome Project, to handle the computing when there are lots of different scales of computing involved, they created the largest computer facility, at least outside of a secret defense industry computer, on Earth at that time, just to do that problem. So when next generation sequencing started popping out after 2001, there were so many reads that you couldn't keep doing what we had been doing. At the beginning, next generation sequencing reads were fairly short, but there were lots and lots of them, and they had substantial errors. So what to do? And here's this famous plot of the cost of 1 million base pairs of sequence going from $8,000 in 2001 to 5 cents today or something like that. So fairly rapid decrease. So, and this is the famous Moore's Law's line. So it sounds impossible to do sequence assembly at that time. And to remind you, all pairs of reads in possible context and so on. So if you had 1 billion reads, 1 billion squared is a number that's a little bit larger to do all of those, even conceptually, all of those overlaps. Because in next generation sequencing, you can have a very deep coverage. So we look back to the 19th century for a piece of mathematics that allowed us to get around this seemingly impossible problem. And my favorite mathematician of all time is Euler, who just invented not quite everything, but almost everything. And in particular, he started a subject called graph theory. And he was incidentally almost blind most of his life, was completely blind in one eye, and then later on the other one got bad. He must have had a phenomenal memory. But the mathematics came from something called the Coningsburgh bridge problem. That you may have seen it in high school textbooks. School kids learn this problem. So there are 7 bridges here. This is the water, in case I'm not clear, and 4 land masses connecting these bridges. And the problem is, could Euler take off and make a walk, get back home, having crossed every bridge once and only once? Now, in the 19th century, people could do arithmetic. So they knew the answer to this problem was no. But what they were really looking at was a mathematical, a general method to look at problems like this and say yes or no. And there are 7 bridges we're trying to have. And if you drew the standard graph that you'd first think of, here's the bridge. It's connected through land masses, like 1 and 3 is connected because they both are on landmass C. And so is there a path that visits all of those nodes once and only once? And that's a Hamiltonian path problem and NP hard. So Euler magically turned this problem inside out and made it into an easy problem. And by that, he made the bridges into edges instead of vertices. And the problem would be to visit all the edges once and only once. It's the same problem. Why would it be easier? Take a look. I'm trying to keep up with the time here. So back here are our land masses and our edges. So the land masses are now the vertices and the bridges are the edges. And I can easily convince you it's impossible to visit once and only once because, for example, let's take landmass B. If we entered on bridge 4 and we left on 5, we would enter on 6 and we couldn't leave. So you need a balance. You need to be able to get on and off that. And if there were arrows, you would have to have as many roads coming in as roads going out, or else you couldn't visit one of these nodes once and only once. So that was Euler's observation, that there's one Eulerian circuit if and only if the graph is connected and the degree is even. They're directed the roads coming in have to equal the roads coming out. There's all kinds of beautiful combinatorics for this. And as I hope to show you in a couple of minutes, once you have one of these Eulerian graphs, finding a tour is really easy. You don't have to be smart. You don't have to look ahead. And I'll just show. So let's flip to sequences, which I'm supposed to be talking about. A de Broglie sequence, our alphabet is 4 letters, as you might imagine. Our de Broglie sequence is a cyclic circle of letters in which all 4 to the K-letter words appear once and only once. And the way to make the graph is the nodes are sequences, the land masses, so to speak, are sequences of lengths k minus 1, and the edges correspond to K-letter words. And I'll show you an example instead of talking more about this. So let's take a sequence and build the Eulerian graph. K is 3 in this sequence, so our first K word is ATG. That is right here, AT, TG. So the first 2 letters, the K minus 1 word is AT, the K minus 1 word here is TG, the edge is ATG. To this point, and the graph goes up here, and then up here, and out. So trust me, or you can scribble, that this sequence would give us this graph. Now, the issue is, if I have the graph, could I get the sequence? Because our assembly problem is going to be taking a graph and reading the sequence from it. So let's just try this guy. This is the graph we just built, supposedly. And the first step, no problem. You have AT. When you do a step, you add the letter. And remember, I promised you didn't have to be smart, but here I have to make a decision. It doesn't matter. Suppose we took this step. We just want to remember there was a path we didn't explore. So we put this exclamation point in and take the step in this direction and get C, etc. And eventually, we'll come back and say, oh yeah, there was a path I didn't explore, and you go back and take those 2 steps, adding a T and a G. So this, whether or not you followed my steps, this is easy. Okay, what does this have to do with sequence assembly? And I stumbled onto this in the early 90s, and absolutely no one was interested until next generation sequencing came along. But then it had to become interesting. So here's the idea. The idea is you've got all of these overlapping reads. And however long they are, we're going to take maybe a K of 25, and we're going to write out all the overlapping 25-letter words and from that read and put them in a data structure. And then if another 25-letter word is identical to one of those, just write over the top of that. You can keep track of how many there are, and so on. So we have this higher dimensional 27 million choose 2 problem that we reduce to a graph having to do with the 25-letter words as edges, and so on. And then if you're lucky and everything's unique, you just, in linear time, read the answer. Now, the wonderful thing about this is there's no pairwise alignment. There's no layout. There's no reverse complement. We handle the reverse complement by every read putting in its reverse complement at the same time. And ideally, you would get 2 sequences instead of 1, and the same time advantage. You don't do any multiple alignment, and for somebody who did alignment 1-letter indels are no worse than 1-letter mismatches. Disadvantages are the graphs are incredibly tangled, and there's a huge amount of storage involved. There are generations of these programs out there now. So that's a place where we had a sequence analysis problem that we had to completely change, and it's changing again today with PacBio and so on. But so now I want to jump to sequence comparison, sequence alignment, which we're getting beyond here today, but there's before next generation sequencing after next generation sequencing once again. And the color changed. Okay, hey, we're on a new topic. So just to go back to sequence alignment, you have the two sequences. These are the aligned letters. The score is the maximum of overall possible alignments of some scoring scheme. And there's a dynamic programming, quadratic time recursion for that everybody knows. Local alignment, which Temple Smith and I were lucky enough to come onto, would be to take all intervals along each sequence, compute the similarity score of those two intervals, and then maximize over choice of the intervals. And that too has this quadratic recursion that has this magical zero added into it. I'm not going through that, but at any rate, that's the algorithm that BLAST is the heuristic for. And you can see this is probably a slide made for an audience less savvy than this. It's so widely used that BLAST is a verb, which you all know that. And the BLAST papers are, a couple of the papers have over 60,000 citations. So this is hugely used by everyone. But clear back in the days when people were doing cDNA libraries trying to discover proteins, the amount of data overwhelmed these more refined alignment procedures. So something called alignment-free, is now called alignment-free, came up, and I'll try to tell you what that is. Essentially, we start with our two sequences, and they have some probability distribution on the letters. P sub L is the probability that L random letters are identical. And D2, which was proposed by David Torney at Los Alamos, is the dot product of the k-word counts from sequence A with the k-word counts of sequence B. So you look at the number of words W occurred in A, multiply the number of words W occurred in B. That's D2. You could change this to a sum of indicator functions by looking at a point in the graph and saying, is there a k-word starting there that's identical at each of the sequences? So you could change this from summing over W to summing over I and J. Here's an example. K is 2, letters are 2, so there's 4 two-letter words. And if you look in A, there are 2 AAs, 2 ABs, 1 BA, 1 BB. And you look in B, and you get 4 ABs and 3 BAs. So the dot product here is 0, 2 times 4 plus 1 times 3, or 11. And we count overlapping, like here, this one's all A's, and so there's the number of AAs is 7, which is 1 less than the length of the sequence. So we emphasize it kind of overlapping. So these two sequences look more like one another. D2 is larger. Gee, maybe this will work. And you'd hope that there'd be a normal approximation for this. If you thought about this in terms of the array, there's an array of zeros and ones, and you're trying to count up the number of ones. So maybe normal approximation works. But in fact, there's a lot of there's dependencies in these overlaps. So there's some very heavy math we can do to check to see that this works. And we did this, and we got a rate of normal approximation to be something, forget what the alpha is, but something that would go to zeros and went to infinity. And yet, no, I should say, what this would say is that a k proportional to logarithm of the length of the sequences with a multiplier of less than 1/6 would have a normal approximation. And yet there was a big problem with our theory, because when the sequences were uniformly distributed, the bound wasn't there. And in fact, it wasn't just theoretically not there, it was not there. Now I'll try to show you why. So this is the only time I really want you to use your brain to try to listen to this. So here we have two-letter alphabet, one-letter words. So we really have none of this overlap nonsense. If X is the number of zeros and Y is the number of ones, X and Y are the number of zeros in the first sequence. In the sequences, D2 is the number of zeros times number of zeros plus number of ones times number of ones. Work it out. You can do the arithmetic, and there's an expected value, but the variance is really interesting. The variance has this form that has a P minus Q in it. So when P is not equal to Q, the variance is big O of n cubed, n squared times n. When P equals Q, this term disappears and the variance is quadratic. So if the alphabet is uniformly distributed, you get this normal approximation. When it's not, it fails. What the heck is going on? And you can go through, I'll just skip this. So I can move on past this. Yeah, so I can kind of show you what we did to fix things. Everybody who took an elementary statistics class, like my students, hears their professor say, subtract the mean, divide by the standard deviation. I hope that sounds familiar. So why don't we do that? We take our word count for W, subtract its mean, and that gives us an equation of at least multiplying centered variables. And if you look at this, you can sort of see where the action is. This is our centered, and then the uncentered part will basically, the variation disappears when all the probabilities are identical, and the uncertainty is there otherwise. So this also explains the previous slide. So to make this work, we had two different methods. One was something that Larry Shepp had observed way back when. If you have two normals, you multiply them and divide the sum of their square roots, you get a normal back. So it's sort of self-normalizing. So we tried for each term W, multiplying these two guys, dividing by their sum. And then we also used a Poisson approximation for the variance. So this is a really different way to do sequence analysis. We're just looking at the K-letter words in the sequence. We're not looking at their global arrangement, how they interact with each other. And this, amazingly, this simple-minded alignment-free stuff does have some applications. That's a lot of applications, actually, looking for gene regulatory regions. In fact, we can do phylogenies and do quite well getting phylogenetic relationships just looking at the K-letter word count, which still amazes me. Metagenomic data, metatranscriptomes, contig binning happens all the time. We are actually working on virus-host interactions, which is a read from a metagenome sample from a virus or from a bacterium. And if it's a virus, what bacteria might they infect? And you can actually learn something about this. And to me, what I just said seems to imply that somehow organisms have control of their K-letter word count. And folks can talk about codon bias and why it's there. That's much easier to think about than how an entire genome can be biased towards some distribution of the fraction of K-letter words. It's totally unclear to me how in the world that happens, and it happens enough that we can learn some biology from it.
Can I ask you a question?
Yes, sure.
So the K, I mean, is this, what is the typical length that as you change?
3, 4, yeah. So that's.
So I'm kind of wondering if you have an assertion, say, you can do phylogenies, right?
Yes.
So is your ability changing with varying Ks?
It depends.
Is it robust?
It's somewhat robust. If you take a really large K, that's pretty useful for doing phylogenetics, really. And you can find something that's unique to that organism. But most of the stuff we do, it's a small K equal 3 or 4 up to 8 in that range. Huh?
What's large?
What's large is 25, say, something like that. Yeah. So there are a few studies. I think we wrote a paper about this. It depends on what, as usual, you focus the microscope depending on what you want to learn. Yeah. Okay. So there's 2 sequence alignment problems. And now we're back to a different color. I'm just going to say a couple words about information theory. And to be honest with you, I did a lot of information theory for my PhD, a bunch of ergodic theory, and I, alas, have forgotten a lot of it, I've realized in the last few weeks. At any rate, the origins of information theory come from physics and from an amazing human being named Claude Shannon, who died not so long ago. And the books you should look at for anybody interested in this, I think his groundbreaking work that is the reason your cell phones work was done in 47, I think, in the Bell System Journal. There's a beautiful book by someone named Brillouin who's trying to struggle with all of this development communication theory versus physics and how all this comes together. And they are linked, and it's deep stuff. There's a book by Lila Gatlin, Information Theory and Living System, which is perfectly naive and atrocious book that had the wonderful feature of David Lippman, whose name you may know from NCBI. David Lippman, while he was a MD PhD student at Columbia, I think, NYU, found this book in Remaindered, and it turned him on to doing mathy things with molecular sequences. So we have Lila Gatlin to thank for David Lippman, which is a big plus. So any book can be valuable. There's just a biography of Shannon just published. The first few chapters are excellent, and then it becomes not as interesting. But the book, if you want to learn information theory today, is Elements of Information Theory by Cover and Thomas. That's the modern book. So here's a figure from that book of information theory and its connections to lots of other subjects. So I'll start here. Communication theory, for sure. Probability theory, especially in large deviations theory. There's a lot you can learn. In statistics, hypothesis testing connects to relative entropy, I think. And a bunch of inequalities. Somebody I just met is doing information geometry and portfolio theory to tell you how to when and how often you should rebalance your portfolio and stock investment. And of course, complexity theory and physics, as I was mentioning earlier, deep connections. Just say a couple of things. The original intuition was by Boltzmann that entropy was K log, disorder was K log n, Y log. And that's easily answered. If you have 2 independent experiments, M in experiment 1 and N, or N in experiment 1 and M in 2, 1 followed by 2 would have M times N states, right? If there are M ways the first one could happen and N ways the second one could happen, the product is the number of ways they could happen together. And cleverly enough, logarithms add. And the constants factor out. So that was sort of how, and I think that was the original combining 2 disordered experiments. And Nyquist brought that from physics to information, as we would normally think about it. Hartley took N and said, oh, if I have an S-letter alphabet and it has N letters, S to the N is the number of states, and so there you'd be. And of course, this adds together exactly properly. The total number of messages would be, if there's a different rate in each one, the multiplication is the right thing. So Shannon brought variation into this, a non-uniform variation, and his entropy calculation is minus sum of p log p, very famously. And for equally likely, of course, it's the logarithm of the number of states. Minus 1 over n, log base 2, 1 over n. Comes out to be log n. And conditional entropy, x if you've seen y, is the p is probability of x and y together, and times the logarithm of the probability of x given y. Mutual information. Essentially, this is the information from y, and this is the information y of x happens. So that's a conditional statement. And I just wanted to show you right here at the end some of the nonsense that was talked about when people were first in biology talking about information. And Lila Gatton's book is related to some of this. Temple Smith and I wrote a little paper. It had been discovered that genes could overlap, that the same region of DNA could be involved in the coding of two different overlapping genes. Fair enough? And we wrote a paper about using information theory to decide what kinds of registers, what kind of shifts would be the most likely to have overlapping coding. And just before us, we didn't know the paper was coming out as ours went in. A guy named Yaki had looked at this problem, and he was looking at the information of frame 1 and frame 2 as frame 1 plus frame 2. And he said, consequently, in practice, to accommodate the genetic code, it must be expected that the sum of the information content of the overlapping sequences will be considered less than the maximum information content of DNA or RNA. And of course, what he didn't realize was that it doesn't add. It's frame 1 plus frame 2 given frame 1. There's no chance of breaking Shannon's theorem because you're encoding two things using the same text. And to me, that's the kind of mistake I wouldn't expect my senior students to make. But there we are. So there are lots of applications of sequence analysis all over the place to align sequences. Califano has done a lot with mutual information and network analysis, discovering master regulators. There's database compression, which has to do with information theory. And there's work that looks at compression and then doing comparison using compression to do sequence comparison a lot faster than BLAST. So information theory is all over the sequences. You know, part of the mistake that people from the outside make, when they hear the word information, they think of what I do when I'm looking for a coffee shop using my internet browser to search. That's information, where the nearest coffee shop is. That's information in the sense that we normally use the word. Information Shannon meant that had to do with the randomness of the text and how to efficiently send messages. You're going to say the same thing all the time, like most college professors do. You don't have to listen all the time. And thank you for listening to me.
We have a little bit of time for questions. Any questions? So I would have one or two, and that would be the idea of this information theory would be centered around the sequence itself.
Yeah.
Right?
Well.
So so kind of, my point would be that it would be the same, that knowing the sequence of the letters to perfection would be knowing the message, right? So there's no notion of interpreting this in different ways.
Well, there's no notion of interpreting. There's a notion of there's a channel. So you have the message you want to send, you encode it, and then you send it through this channel which has noise, and Shannon can tell you how to compute the maximum amount of information you can send through a given channel. And he also says you can come arbitrarily close to that if you're willing to repeat noisy messages a lot. And so at the other end, you're supposed to decode, you're supposed to know how the encoding went here so you can do the decoding.
Right. So you get theoretical bounds on what information you can transfer.
Channel capacity information, yes.
So I wonder about the channel now.
Yeah.
Right? I wonder about the method of encapsulating that piece of information, right?
Yeah.
Like there are theoretical limits as to what I can
Yes.
transfer, and say I'm below those limits, what are my benefits of, right? Do I get more redundancy? Do I get more fault tolerance?
Essentially, it's not quite law of large numbers, but repetition is what's doing this, right?
Right.
And of course, and part of his thing is you're going to say you can just send one of those and then try to indicate how many times.
So coming back to the K-words. So say you're in a non-coding region of DNA. Then if you look at the K-word, you're somewhat local because you say you go say 20 nucleotides. Yeah. But we know that, for instance, if a structure is formed,
Sure.
we have a helix.
Yep, absolutely.
And in helix, you would observe base pairs. And these base pairs, they could be far apart, far bigger than 25.
Yeah.
So it's kind of remarkable how descriptive the K-words are, given that they are so local.
No, and the different stuff is happening in different parts of every genome. No, it's
right, because I would have, in a sense, it would be really nice if you could guess the gaps, and then have instead of having a singular K-word, having, let's say, a pair of K-words, start guessing the gaps that you would have to bridge in order to see the correlation between these positions. Right, are there any questions? Yes?
So can I ask one about the phosphate K-word thing?
About what?
So
Yeah.
is that like the K-words you can discriminate things between species? Can you give me a concrete example for that? So what from what?
So I think you're asking in this phylogenetics, do we pull out the K-words that are really where the action is? Is that, yeah. And we haven't been very successful at doing that. So it's somehow a more global assessment. And also, even though I made my living on the statistics of words that are overlapping or non-overlapping and so on, self-overlapping and none of that comes into these alignment-free calculations. So it's a very crude measure.
All right, are there any more questions? Otherwise, I think the speaker again.